Introduction: CRISPR gene editing has revolutionized genetic medicine, offering new possibilities for treating genetic disorders, enhancing agricultural productivity, and even combating infectious diseases. The development of CRISPR, clustered regularly interspaced short palindromic repeats, technology has significantly accelerated biomedical research and therapeutic applications. This article explores the science behind CRISPR, its applications, ethical concerns, and its future in genetic medicine. What is CRISPR? CRISPR is a revolutionary gene editing tool derived from bacterial defense mechanisms against viruses. Scientists discovered that bacteria use CRISPR-associated proteins, Cas, to cut viral DNA, preventing infections. The most commonly used Cas protein in genetic editing is Cas9, which allows precise modification of DNA sequences in living organisms. How CRISPR works. 1. Guide RNA, gRNA, identification: a designed RNA molecule that directs Cas9 to a specific DNA sequence. 2. Cas9 protein activation: Cas9 acts as molecular scissors, cutting the targeted DNA. 3. DNA repair mechanisms: the cell repairs the cut DNA, either disrupting a gene or incorporating a new genetic sequence. Applications of CRISPR in genetic medicine. CRISPR technology has numerous applications in medicine, agriculture, and biotechnology. Below are some of its most promising uses in genetic medicine. 1. Treating genetic disorders. CRISPR offers potential cures for genetic diseases by correcting faulty genes. Some key applications include: 1. Sickle cell disease and beta thalassemia. CRISPR has been used to edit blood stem cells, restoring normal hemoglobin production. 2. Cystic fibrosis. Scientists are working on correcting the defective CFTR gene responsible for the disease. 3. Huntington's disease. CRISPR can target and silence mutant genes that cause neurodegeneration. 2. Cancer therapy. CRISPR is being used to enhance cancer treatment by 1. Editing immune cells. CRISPR can engineer T cells to recognize and attack tumors more effectively. 2. Targeting oncogenes. Researchers are disabling cancer-promoting genes to halt tumor growth. 3. Infectious disease treatment. CRISPR technology has shown potential in combating viruses, including: 1. HIV. Researchers are using CRISPR to remove HIV DNA from infected cells. 2. Hepatitis B. CRISPR has demonstrated effectiveness in eliminating the hepatitis B virus from liver cells. 3. COVID-19. Scientists have explored CRISPR-based diagnostics and antiviral therapies. 4. Organ transplantation. CRISPR enables genetic modifications to make animal organs more compatible with human transplantation. This breakthrough could address organ shortages and save countless lives. 5. Personalized medicine. CRISPR supports personalized medicine by tailoring treatments based on a patient's genetic makeup. This approach enhances drug effectiveness and reduces side effects. Ethical concerns and challenges. Despite its promise, CRISPR raises several ethical and technical challenges. 1. Off-target effects. CRISPR may inadvertently edit unintended DNA regions, leading to potential risks such as mutations or unintended consequences. 2. Germline editing. Editing germline cells, sperm, eggs, embryos, can result in heritable genetic changes. This raises ethical concerns regarding designer babies and unintended long-term effects on future generations. 3. Accessibility and equity. CRISPR-based treatments may be expensive, creating disparities in healthcare accessibility. Ensuring global access to gene therapy remains a key challenge. 4. Regulatory and legal frameworks. Governments worldwide are establishing regulations to govern CRISPR research and applications. Ethical guidelines are essential to prevent misuse and ensure responsible development. Future of CRISPR in genetic medicine. The future of CRISPR is promising, with ongoing advancements expected in: 1. Next-generation CRISPR systems improve precision and efficiency, reducing off-target effects. 2. CRISPR-based drug development targeting diseases beyond genetic disorders, such as Alzheimer's and diabetes. 3. Synthetic biology applications designing new biological functions and enhancing human capabilities. Conclusion: CRISPR gene editing is shaping the future of genetic medicine by offering unprecedented possibilities for disease treatment and prevention. While ethical and technical challenges remain, ongoing research and responsible use will determine how this powerful tool transforms medicine. The potential for CRISPR to revolutionize healthcare is immense, making it one of the most significant scientific breakthroughs of the 21st century. By continuing to explore and refine CRISPR technology, we move closer to a future where genetic diseases are no longer a major health burden, and personalized medicine becomes a reality for all. Subscribe.